Hello everybody, so this is the Separates Biology Paper 1 walkthrough for the 2024 paper. Okay, so this is a nice easy one to start with. A person has coronary heart disease. Which blood vessels are affected by coronary heart disease? Okay, so the only answer for this is, of course, arteries. So a person's heart stops beating. A person stops breathing. A first aider pushes down on the person's chest. Pushing down on the person's chest puts pressure on the heart. Explain why putting pressure on the heart helps the person. Now, there are a number of ways that pupils have answered this question incorrectly. So they start talking about restarting the heart. They start talking about pacemakers. They start talking about kind of CPR and the process of it. But that's not what it's asking. It's asking, why does putting pressure on the heart help the person? So what we're looking for is for you to mention that we are pushing or moving the blood, and that this carries oxygen around the body or to the cells. Okay, now because it's only 2 marks, you don't have to link it to respiration quite here. All right, you could talk about oxygen getting around the body through blood vessels or to get to a named organ. So, for example, to get to the stomach or the liver or the muscle cells. The only organ that you couldn't really actually include in that is the lungs, because obviously you don't. It's not to get the blood to the lungs in this case because the person is not breathing, so it wouldn't matter. So there's your answer. So the first aider forces air into the person's lungs by blowing into their mouth. Describe how forcing air into the person's lungs helps the person. So this is where we need to start saying, all right, well, this is where we are providing gas exchange. We are providing oxygen for respiration. So that would be the answer there. So it's to provide oxygen or to give oxygen for respiration. Now, to be fair, if you haven't included for respiration, you still get the mark at this point, and this is because it's really early on in the test. However, I would always say that if you are mentioning oxygen, we've talked about this in lesson, you absolutely need to be talking about respiration. So the next question, the person's heart starts to beat again and the person starts breathing. The person has a high level of cholesterol in their blood. Name one type of drug that would decrease the level of cholesterol in the person's blood. So the only answer to this is a statin. Now, if you've got a member of your family who takes statins and you know either the brand name or the actual chemical name, that would be accepted as well. So the most common statin is atorvastatin, then there's simvastatin, fluvastatin, provastatin, rosuvastatin. Essentially any drug that ends in the word statin is a statin. All right, so they would all be acceptable answers as well. Something I do actually have to mention on the 1.3 that I completely forgot to talk to you about is the fact that if you exhale carbon dioxide into somebody, because obviously when we are exhaling, we do exhale more carbon dioxide than we breathe in. That presence of carbon dioxide can sometimes be enough to trigger the breathing to restart. So if you've mentioned that for 1.3, well done. You can have that question as well. A doctor decides that the person needs to have a stent fitted. Explain how a stent works to treat coronary heart disease. Okay, so what we're looking for is that it is opening or widening the blood vessel. All right, now I do need to see that word. Okay, I do need to see that you have widened a blood vessel. Okay, really important. Or that you are mentioning that it keeps the vessel open. Or it might be description. So if you've described the whole process where you've got the little metal cage, if you will, the actual stent that you put in, and then there's a little balloon that inflates it and it kind of opens it up, that's fine as well. Unblocking the blood vessel is not correct though. So if you said unblocking, that is not correct. It has to be opens or widens. And then that's the description of it. And then the explanation is, how does it work to treat coronary heart disease? Well, it allows more blood to flow. Okay, or just blood to flow. You don't have to say more there. All right. Or you can say, link it back to respiration, because that's always a good thing. You could say, oh, it allows more glucose or oxygen to flow to the heart cells, tissues, muscles, all that kind of thing. Okay. Okay, so we have this table, and we've got a table showing the effect of smoking on the risk of developing different cardiovascular diseases. Okay, so you've got cardiovascular disease E, F, G, and H, and then the percentage increase in risk compared to people who've never smoked. So just to be aware, these aren't people that we're talking about. These are different diseases. Okay, so commonly in the past, pupils have gone, oh, person E is more at risk, and that's not what it's saying. Okay, so these are 4 different cardiovascular diseases, and it's looking at, okay, comparing the increased risk to somebody who has never smoked. So for cardiovascular disease E, if you've never smoked, you are 14% less likely to have the disease. But if you have smoked, you are 14% more likely. With F, if you've smoked, you are 20% more likely to have the disease than somebody who's never smoked. G, 29%, and H, 70%. So some conclusions that can be made from this data. Well, as I've just said there, the most obvious one is that smoking increases the percentage risk of all types of cardiovascular disease. So smoking increases risk of all. I'm just going to put CVD, okay, because I'm running out of space. Cardiovascular disease. So I'm just going to call that CVD up there. Okay. I would then say another one would be the fact that that's the biggest one. So I say that smoking increases the risk of H the most, or more than any other type of disease. Or if you smoke, you are most likely to develop disease H. And then another thing you could say is that you are least likely to develop E, or smoking increases the risk of having E less than any other cardiovascular disease. So you would say less risk than other cardiovascular diseases. Okay. So not smoking, and you could conversely say these things as well. So instead of saying smoking does this, like I said at the start, you could say that not smoking decreases the risk of all of the cardiovascular diseases. And there you are, your answers. So again, please make sure that you have recognized that these are 4 different diseases and not 4 different people that you are talking about. If you start talking about people, you will have lost all of the marks. There is no marks that you can get. Okay, so this is where you've been asked to then make a bar chart of the data that was provided. So I've re-put table 1 there. Now, you get 4 marks for this. Okay, so you get 1 mark for, and this is why it tells you certain things. You get 1 mark for labeling the axis, 1 mark for scaling, 1 mark for drawing the bar charts, and then 1 mark for labeling each bar. I mean, to be fair, there's not many times that pupils don't get the full 4 marks for this, but it normally comes from mislabeling or not labeling the y-axis. Okay, now this has already been labelled on the x-axis. Therefore, we need to put percentage increase in risk. Okay, now you could just leave it at that. That is absolutely fine. Percentage increase in risk. Now, if I was you, I would write the whole thing. I'm not going to do that today because I'm going to run out of space very quickly if I do that. But percentage increase in risk compared to people who've never smoked. Then you need to add the correct scale to the y-axis. Remember, this is the y-axis. Now, for me, I do 0. I would then do 10, 20, 30, 40, 50, 60 and 70. Of course, you will have done this in pencil and you'll be using a ruler. A bit difficult for me, but I'll try my best. Okay, now the reason I've done that is because E has already been done for us, which is 14, and that's pretty obvious that that's 14 there because you've got the 10 line here, okay, and then we've got the 14 line there, which is just 1 down from 15, so that makes absolute sense. Okay, now We then have to plot the data from table 1. Okay, so E's been done for us. So F is going to be at 20. And again, you are going to do this with the ruler and you are going to label it there as F. G is going to be 29, which will be here. And again, that will be labeled G. And then H would be 70, which would be all the way up here, like so. And again, you will be using a pencil and a ruler. Okay, you don't have to shade it in. Okay, so as an examiner, the amount of times I see people who've done lovely little cross hatchings and shadings in, and like, you don't get marks for that and you're wasting your time. Okay, just 3 bars like that is perfectly fine. And we've done all those 4 things. We've labeled the y-axis, we've added the correct scale, we've plotted this data, and we've labeled the bars. Okay, so double check every time that you get a graph, whether it's a line graph, whether it's a bar chart, what the 4 bullet points will say, or the 3 bullet points will say, is going to get you the marks. And if you ignore that, you're just throwing away marks. When you are asked to plot a kind of line graph, so you do your x's and then like you do a line of best fit, you will always get 2 marks for plotting those points, providing there's more than 3 points. With a bar chart, because bar charts are seen as a much simpler graph, you'll only get the 1 mark for drawing all 3 bars. Describe one lifestyle factor that can increase the risk of cardiovascular disease. Do not refer to smoking. Now, we can't refer to smoking because you've just had a whole graph that clearly shows you that smoking increases the risk of cardiovascular disease. So if it's asking you not to refer to something and then you go and refer to it, make it make sense. Okay, so what we're looking for here is anything along the lines of poor diet. Now, I don't like poor diet because sometimes they'll accept poor diet and sometimes they won't. Because it's in question 1, they will accept poor diet. However, I would like some meat on the bones of what that poor diet means. So I'm looking for a diet in saturated fats or cholesterol. So it's high in fats, it's high in cholesterol. Okay, if you've written poor diet, because it's on the mark scheme as allowing that, I will allow it for you, because obviously that is a mark point. However, what I would like you to do is add to your notes as we're going through this that you need to make sure that you are writing things like high cholesterol and high fat in the diet. So first thing is poor diet. Okay, so we're looking for high in fat or cholesterol. All right, and then another one could be lack of exercise. Okay, lack of exercise, which is pretty self-explanatory, I think. You could also have things like high alcohol content or high alcohol intake, or you could say such things as having a stressful job. Okay, those are kind of things as well. Obesity is not a risk factor. Okay, now the reason being that is because obesity is the kind of culmination of other lifestyle factors, if that makes sense. So obesity is kind of like the end point of lifestyle factors that have led up to that. Okay, so such as a diet high in fat and not having any exercise. Cystic fibrosis is an inherited disorder caused by a faulty gene. Where in the cell would the CF gene be found? Now it's asking where in the cell, so you need to think, right, where in the cell do we find genes? Do we find DNA? That is obviously the nucleus. Now, we have spoken in the past how they can pull your knowledge of paper 2 onto paper 1 and paper 1 onto paper 2. So we've talked about cystic fibrosis and inherited disease in paper 2. Okay, so again, there is that kind of link there. So don't be frightened when you see things like CF on paper 1, sorry, that is from paper 2. CF affects many organs in the body. The main organs affected are the lungs, the pancreas, and the small intestine. Okay, so even though this is a paper 2 topic, it's leaning into your knowledge of paper 1, which is the digestive system, which is organization. So figure 2 shows organs of the human body. We've got A, B, C, F, E, and D all labeled for us there. Which letters show the lungs, the pancreas, and the small intestine in that order? So there are the lungs, that is the pancreas, and that is the small intestine in that order. So it's A, D, E. B is the stomach, C is the large intestine, and F is the liver. Okay, so you've got the liver and then you've got the little gallbladder there. The pancreas produces several digestive enzymes. CF reduces the amount of each enzyme that reaches the small intestine. Explain why a person with CF has difficulty digesting food and difficulty gaining body mass. Okay, now this is a leveled question. Okay, so in order to get the full 6 marks, you have to talk about the difficulty in the digesting food and why they would not gain body mass. If you haven't mentioned why they would not gain body mass, you can get a maximum of, is it 2 marks? 3 marks? Let me just have a quick check. No, you can get a maximum of 4 marks. Okay, so if you don't mention this, okay, so look at your answer when we go through it in a second. If you've not mentioned difficulty gaining body mass, maximum mark you can get is a 4. Okay, regardless of what you said. Okay, so we're going to do the difficulty in digesting food first. Okay, so we'll do the digesting food bit first. All right, so the reason they've got difficulty in digesting food is they have got less lipase. Okay, so less lipid broken down into fatty acids and glycerol. Now, here's the thing. If you've just written that, you're automatically on 3 marks, okay? And this is why. You've given me a point, which is the less lipase, and then you have linked it to the difficulty in the digesting foods. This is what we call a linking mark. So you're already into that 3, okay? So well done. Now, the other ways that you can do this, okay, is by saying, or how you would get the extra 4, get the 4th mark if you've not mentioned the difficulty in digesting body mass, difficulty in gaining body mass, is that obviously we could talk about the fact there's less amylase, and then we can talk about the fact there's less protease. Okay, so if you've just said less lipase, so less lipids broken down to fatty acids and glycerol, and there's also less amylase, you've already got 4 marks. That's it, done. If that's all you've written, 4 marks. If you said less amylase, so less starch broken down into glucose, so less starch broken down into glucose, okay, or you've said less protease, so less protein broken down into amino acids, okay, you'd also get just the 4 marks. However, you could just say this bit plus one other bit there, this bit plus one other, or this bit and plus one other. If you've written all 3 of those things, but you've not mentioned difficulty in gaining body mass, you still get maximum 4. Okay, nothing we can do no matter how much you've done. However, if you've just said less lipase, so less lipid being broken down, so less lipid being broken down, but then you stop there, or you said less amylase, so less starch being broken down, and then you stop there, you are only going to get a maximum of 2 marks, okay? Because you've not done what we call a linking part, okay, which is the linking to the full complete digestion. So you need to make sure that you've gone through the full digestion process. Okay, now, difficulty gaining body mass, which is historically where pupils have lost marks on this one, okay, from when I've looked in the past. Okay, so they've got difficulty gaining body mass because they're not going to have fatty acids and glycerol, glucose, amino acids. So there's going to be less absorption of fatty acids, glycerol, sugar, amino acids. Okay, less absorption of those things. So then you could say, so then there's less. So if we link it to here, there's less respiration. Okay, that would be an issue. With amino acids, there's less proteins being built. So less proteins built, less fat stored. Okay, and all of these are to do with the difficulty in gaining mass. Okay, So again, if you've not mentioned gaining mass, but you've done the bit about digestion really well, maximum marks you can get is 4. So please make sure that if you are told by the examiner, you're told by the exam paper to include something, that you include something. Okay? Gas exchange happens in the alveoli and the lungs. Describe 3 features of the alveoli that help maximize gas exchange. Okay, now it's just a description, okay, and it's only 3 marks. So it is a big 3, but it is literally just stating what the big 3 are. It's not linking the big 3 to how the alveoli are helping with the maximizing of gas exchange. So large surface area, obviously. And then we've got good blood supply or large capillary network, many capillaries. From what I've seen in the most recent exam is that they are wanting you to mention the word capillaries. Okay, so make sure that you are doing that. All right, so good blood supply, but what we really mean by that is that they have got lots of capillaries or covered in capillaries. And then now remember this one, the walls are thin or 1 cell thick. Okay. Alveoli are thin. Alveoli are 1 cell thick. No. Okay, it has to be the walls of the alveoli are 1 cell thick. Any reference to membranes? No. Okay. Anything to do with them being moist or to maintain that steep concentration gradient? No, because that is an effect of the other things that we've talked about. So CF reduces the amount of oxygen that can enter the blood from the alveoli. Explain how a reduced amount of oxygen entering the blood will affect the human body. Okay, so straight off, when we see the word oxygen, we need to link it to respiration straight away. So that should be the first thing that you go to. So the first thing is we've got less respiration. So we've got less energy released. And then we need to think about what is that energy used for. So straight away, I'd be like, right, less muscle contraction. And that straight off is 3 marks. Okay, nice and simple. Now, you could say another thing is there's less energy released, so a reduction in metabolism. Okay, that's absolutely fine. You could also then mention some other things. So you could say an increased breathing rate. Okay, so if somebody's getting a reduced amount of oxygen, we've got a reduced breathing rate. Okay, or you could say that they're getting out of breath more. Okay. You could also say that there is an increase in heart rate as well because there's less oxygen into the blood, so the heart is pumping to get that blood around get that oxygen around the blood faster. But either way, it's all linked to this idea of less respiration. Now, conversely, particularly those that you do sport, you might think about it from this way: is that you might say, well, actually, there's less aerobic respiration, but there is an increase in anaerobic respiration. All right. Now, an increase in anaerobic respiration would mean more lactic acid produced, so more muscle fatigue. So therefore, we've got increased lactic acid, which is equal to increased fatigue of the muscle. Okay. Or you could say there's less muscle contractions because there's less energy released. And then you could talk about oxygen debt. So you could say, well, the end result would be they have an increased breathing rate and an increased heart rate because they're trying to remove that lactic acid that is built up. Okay, so there's 2 different ways that you can approach this question. Talk about less aerobic respiration, or you could talk about increased anaerobic respiration. Okay, so historically this question's been really poorly answered. Okay, so cake and bread each contain the same 2 types of carbohydrate. So there's 2 there. So first off, we need to identify that it's starch and sugar that we're talking about. Then we need to describe the presence of the 2 tests. Okay, so we need to look at the 2 tests and then include a risk assessment. If you have not included a risk assessment, then the maximum mark you can get is a 4. Okay, so again, like we were talking about before, if you don't include things that the examiners asked you, you're being silly, you are just throwing marks away. Okay, so this is how I would set it up, right? I need to do the 2 tests. So I need to do starch, I need to do sugar, and then I need to do my risk assessment. Okay, so straight off in my head, I know the things that I need to talk about. So starch, I'm going to test using iodine, and I'm going to say that orange is negative and blue-black is positive. Now, I've done shorthand for negative and positive there. You do not do that. You need to make sure you write it out in full. You don't want the examiner to be like, do they really know what they're talking about? Do not give them that option to take marks off you. Now, sugar, we're going to mention Benedict's. And Benedict's is we're going to use 95 degrees Celsius for 5 minutes in a hot water bath. And again, I've written HWB. You are going to write hot water bath. We're then going to write red for lots, yellow, orange for some, and we're going to put green for little, and we're then going to put blue for negative. Okay, now we need to talk about the risk assessment. Okay, so what are the risks in this? All right, so risks of burning from the boiling water. So you're going to have boiling water scalding or burning. Okay, so what do we use? Well, we use the hot water bath. Okay, so we use a hot water bath, or we allow the glassware to cool before we touch it. Okay, another thing you can say is that Benedict's is an irritant, so is iodine. Okay, so these are both irritants. So what do? We wear goggles or safety specs. Okay, so it's not just enough with a risk assessment, it's not just enough to identify the risk. You also need to say how are you going to mitigate or how are you going to reduce the risk to you. It's really important. You could also say the fact that you could use a dropper bottle to reduce the chances of spills, clean up spills immediately, point test tubes away from you when you're heating things, all these kind of things. Okay, anything that's reasonable. If you're unsure about what would be accepted, if you want to bring it to me, then we can have a quick look at it. All right, but it's mostly, it's pretty much self-explanatory. Okay, so here we have an investigation where a student has investigated 3 different types of bread. For each type of bread, the student has put a square piece of bread into their mouth. They haven't chewed it, and they've recorded the time taken for the bread to start to taste sweet. And table 2 shows results. So you've got 3 types of bread: brown, white, and wholemeal, and then you've got the time taken for the bread to taste sweet in seconds. So the brown is 43 seconds, white is 35, and wholemeal is 57. And it's asked here, what is the dependent variable in this investigation? So remember, the independent is what you change. So I am changing that because it's the independent. The dependent is essentially what you are recording during the practical, and then the control is what stays the same. Or for me, what I always think of is the independent is the left-hand column and the dependent is the right-hand column. So in this case, if we're to do that, the right-hand column is the time taken for the bread to taste sweet. Okay, now time unqualified. So if you've just written time, then you would not get the mark. It has to be time taken for bread to taste sweet, or time taken to become sweet, or to turn sweet, anything like that. I'm going to say turn sweet. Okay, so taken to, turn to, whatever. Give 1 control variable the student should have used in this investigation. So this is what they needed to keep this kept the same. Now, size or mass of bread, that's 1 mark. Okay, so that's what I would have said. Size of piece of bread or the mass of bread. Amount of bread is allowed. I don't like it because it's not very scientific, but they're allowing it. Okay, I would prefer to see mass. The surface area of the bread is another one you could say. The location of the bread on the tongue or the position of the bread in the mouth. Or you could say the fact that you need to clean your mouth out between tests. So you do the brown one, then you spill your mouth out with water, then you do the white one. Anything to do with the temperature of the bread or the same student? No, ignoring those ones. Okay, so it's just the ones that I've said. During the investigation, the bread began to taste sweet in the student's mouth. Explain why that is. Okay, so first thing you need to say is that the bread contains starch. Okay, you should know that bread is a starchy food, okay, and it contains starch. We then need to say that it is broken down by amylase into glucose or sugar. Okay, and that's your 3 marks. So you get bread contains starch broken down by amylase into glucose and sugar. Now, it doesn't have to be in that order. You could say that the bread contains starch that is broken down into glucose by amylase. It doesn't really matter. You could say the bread contains, another alternative way you could say is that bread does actually contain sugar and glucose. So you could say that the bread contains sugar and glucose. It dissolves in the saliva that's picked up by the taste receptors on the tongue. So there is a different way of talking about sugar. The most obvious way is that your saliva contains amylase. That's what's happening. But the other one is talking about the sweet receptors on the tongue, and they are stimulated by the sweet sugar that is already in the bread that has been dissolved by the saliva. Suggest one reason why the results in this investigation were not valid. Do not refer to control variables. So I don't want to see anybody mentioning anything to do with the bread's not the same size, etc., etc., because we don't know that, because it's not there. So we're going to assume that there are different reasons why this is not valid. And the first one that I would always go for, as with any kind of practical or any kind of experimental data, is there is no repeats. All right, so we've got no repeats by the same or different students. Because we're relying on taste is subjective. Okay, so and we know this because some people, you can eat the same foods and be like, oh, that's really sweet. Some people are like, that's not sweet at all. Oh, this is really spicy. It's not spicy at all. So it relies on the student's perception of taste. And the amount of saliva that people produce is also variable as well. Okay, I would always go for there's no repeats with the same or different students. But like I said, there are a couple of different things that you could say as well. Anything to do with referencing to your control variables? Absolutely not. Anything to do with if you've just gone, there's no mean being calculated. Well, you can't calculate a mean on one result, can you? So you have to make sure that you've mentioned the repetitions. If you said, make sure there's repeats so you can calculate a mean, which this one is missing, that's fine. Okay, but if you've just said repeats for a mean, just be very careful with what you're saying, that it doesn't leave you open to interpretation, or the examiner's a bit like, oh, I don't really know what they mean by this. Plants contain many different tissues. Complete the sentences. So the leaf tissue that contains the most chloroplasts is the palisade. So the palisade layer is the layer that's right at the top of the leaf. The leaf tissue that contains many air spaces is the spongy mesophyll. And the plant tissue that can differentiate throughout the entire life of the plant is the meristem. So those are your only answers. There's no other kind of way that you can say this. Xylem tissues transport water through a plant. The walls of the xylem contain cellulose. Name one other substance that strengthens the xylem tissue. And again, only one that we know of, and that is lignin. Phloem tissue transports dissolved sugars around a plant. Name the process that transports dissolved sugars around a plant. So this is translocation. Okay, now on the exam, some pupils started talking about active transport. Now that could very well be how nutrients get into the root hair cell, but it's not how they are transported around the body. That is translocation, which is dissolved sugars. So here we have a picture that shows two plant cells. We've got the phloem cell, and then we've got cell X. Name part Y in figure 3. Okay, so that is clearly a large space that is inside the plant cell. So that is a vacuole, and that is the only answer that you can give for that. Phloem tissue transports sugar to other parts of the cell. The concentration of dissolved sugars in the phloem cell in figure 3, which is the one that we've just looked at, is higher than in cell X. Explain how subcellular structures help to move dissolved sugars from cell X into the phloem cell. Okay, so it's already said that it's higher. So the phloem cell, the concentration is higher than in cell X. And it's asking us how we go from cell X into the phloem cell. So we are going up a concentration gradient. Okay, so straight off, we need to be thinking this process, because it's going up a concentration gradient. So sugars are being moved against a concentration gradient. All right, and then we need to define the process for this. I was going to put conc gradient. Again, you make sure that you write the full thing. Use via the process of active transport. Okay, and then we move back to, right, okay, what is required for active transport? Okay, so we now need to link it to this part of the question here, which is asking about subcellular structures. Now, really, there's only one subcellular structure we need to talk about. Active transport, this requires energy to be released from respiration, which takes place inside the mitochondria. And there we've hit all the mark points. We've mentioned the subcellular structures. We've mentioned the process that which the dissolved sugars are moved against concentration gradient. Again, essentially, it's just understanding our or expressing our knowledge of active transport in a slightly different scenario. So I've got the picture of figure 3 again, because we need to refer to this in this question. So the new phloem cells form when unspecialized plant cells differentiate and become specialized. Bit of tongue twister that. Describe one change in structure that occurs when an unspecialized cell differentiates to form a phloem cell. So essentially, we're looking at cell X, we're looking at the phloem cell, and we're going, what is the difference? Okay, describe one change. So basically, we are just saying what is happening. It's very much catchphrase. It's very much say what you see. All right, so there are loads of things that you can say. Okay, you will need to say one of these. Loss of cytoplasm, loss of nucleus, loss of mitochondria, loss of ribosomes, or altogether under the umbrella term of loss of subcellular structures. You can say that the end walls have become perforated, okay, or sieve plates have formed. Cell walls have formed, larger vacuole forms. Okay, that's it. There's loads of different things, but that is essentially it. I would have gone for, I'd probably just gone for loss of subcellular structures. So loss of subcellular structures, because you can see that. I mean, look at how complex cell X is in comparison to the phloem cell. I mean, look at this. You've got so much stuff in here, and that is essentially an empty cell, okay, that is essentially mostly xylem. It's not. It's mostly vacuole. Okay, so this is a big 6 marker, but a bit of a gift of a 6 marker. So we're given this graph where a student is investigating the effect of concentration of salt solution on the mass of uncooked potato pieces, and it's giving us a very, I would say, unique graph that we only see once in biology, where we've got positive and negative percentage mass, mean change in mass of potato, and then we've got a concentration. So straight off, this is an osmosis practical. So this is asking us to set up an osmosis method. Okay, and it's saying that we're using it on potato pieces. Now, in the past, like I've said to you in class, I've seen this on beechworms, on eggs, on boiled carrot, etc. It does not matter. It's always the same thing. So first thing that I would be saying, okay, is to cut out, cut out and record the start mass of, how many different concentrations have we got there? Well, it doesn't really matter, does it? Should we say 5? We'll say 5 pieces of potato. So cut 5 pieces, not like an S, but it is a 5 pieces of potato and record start mass. Okay, you're then going to place potato, so place each piece into, and we'll say 25 mils, because it's always nice to put volume, of different salt concentrations, and then we will say 0, 0.2, 0.4, 0.6, 0.8, and 1. Oh, actually, maybe 1, 2, 3, 4, 5. 6 pieces of potato. Right, 6 pieces of potato. Okay, in different salt concentrations. Okay, and they're the salt concentrations. We're then going to leave for 24 hours. Then we're going to 24 hours. Then we're going to blot and reweigh. Okay, so we're going to blot and reweigh. We're then going to calculate the change in mass. Okay, and that'll do nicely. Okay, calculate change in mass, and then maybe it actually calculates mean change in mass because we're going to repeat 3 times at each concentration. Okay, now within this, we also need to mention some control variables. Okay, so we could say 6 pieces of the same potato. That's another way of saying it. We've got the 25 mils of the different concentrations. That will do nicely. So we've said leave them for 24 hours as well. So that's another control. Okay, happy days there. As an examiner, somebody presented that with me, I'd be like, congratulations, 6 marks. Okay, however, in order to get into level 1, it must allow the determination of the change in mass for a piece of potato. So if you have not mentioned weight before and then weight after, you can't even get level 1. Okay, so just be wary of that. Okay, for level 3, which is your 5 and 6 marks, it must allow the production of the graph in figure 4. So you need to mention the concentrations. You need to mention calculating the percentage mass, the mean change in mass. Sorry, not the percentage mass change, the mean change in mass. But to be fair, I think percentage mass change would probably be okay as well. But it's not percent, is it? It's mean change in mass. So they've not done a percentage mass. They've just done the mean change in mass. Okay. Okay, so we've got this graph again. A student investigate the effect of concentration on salt solution on the mass of uncooked potato pieces, and then it says explain the results for potato pieces in the 0.6 mole solution. So we look at 0.6 and we go, right, okay, it's here. Okay, so it has lost mass. Okay, in fact, it's lost 0.2. Okay, so what we need to say here is why has it lost the mass? It's lost mass because water has left the cell via osmosis. Okay, so straight off we've got 2 marks there. Lost mass because the water has left the cell via osmosis. Okay, and then we need to say because the solution in the potato cell because the solution in the potato cell is more dilute than outside. In the cell is more dilute than outside the cell. Or you could say because the solution outside is the solution in the cell is less concentrated than outside. So you could say that as well. Or you could say the solution outside the cell is less dilute. Anything kind of that. If you have written water moves from an area of high concentration to an area of low concentration, because it's not, is it? If you said from a high water concentration to a low water concentration, that is fine. So if you have said lost mass because water has left the cell via osmosis because water moves from an area of high water concentration to an area of low water concentration, that is fine. Or you have said it is moved from a dilute solution to a concentrated solution, that is also fine. Explain why the results for the potato pieces at 1 mole was different than at 6. So at 1, okay, it has lost more mass. Okay, so 1, it's lost more mass because more water has left. So loss of more mass as more water has left. And what we need to say here is because there is a greater difference in concentration or a steeper concentration gradient. Now, when we're talking about the steeper concentration gradient, like I've said, so you need to say there's a greater difference in the concentration between inside and outside the potato at 1 than there is at 0.6. Okay, so it's this idea that at 1 outside the cell it is more concentrated. So if you said it is more concentrated outside the cell for 1 than it is for 0.6, that is fine. The solution is more concentrated. That is fine. That would be your second mark. Okay, you could also say more mass has been lost because more osmosis has occurred. That is also fine. So with that top bit, loss of more mass as more water is left, or you could say loss of more mass because there's more osmosis occurring. That is also fine. So, number 6, moving on to infection response now. This question is about pathogens. A scientist investigated antibiotic resistance in bacteria. Name one antibiotic now. There are hundreds of antibiotics. Obviously, the most common one is penicillin. Okay. However, I would accept anything like amoxicillin, streptomycin, chloramphenicol. All right. Essentially, anything that you have been described. So, as an examiner, if somebody's written the name or a brand name or a name that is not penicillin, I will always go. So if I know it from having worked in the NHS, I'll be like, oh yeah, cool, that's fine. If I'm not, I'll always give it a quick Google, be like, is this an antibiotic? You know, and it happened. This actually happened when I was marking this as an examiner a couple of years ago. There are more antibiotics than they can list in the mark scheme, so it's fine. However, really common issue is that pupils write things like painkillers or ibuprofen or aspirin or things like that. And remember, antibiotics are targeted at bacteria to treat bacterial infections. They do not treat symptoms such as cough and cold medicines, aspirin, etc. A student grew one type of bacterium in agar in a petri dish. The scientist placed paper discs, each containing a different antibiotic on the agar, and it shows the appearance of the petri dish after 2 days. So we've done something similar to this. Okay, so the bacteria are growing on an agar quite nicely, fully covered the agar, and then they've put 3 different antibiotics on. So they've got antibiotic disc A, B, and C. Now, the student said the bacterium is resistant to antibiotic C, which is this one here. Okay, explain how the results show the student is not correct. Okay, well, it's not correct because you've got a zone of inhibition around that antibiotic C, haven't you? Okay, so there is a zone of inhibition or the bacterium has been killed. So the bacterium or bacteria have been killed around C. Alternatively, you could say zone of inhibition. Okay, that's absolutely fine. Same mark point. And then you have to basically say, if it was resistant, there would be no zone of inhibition, or the bacteria would not be killed. Okay, so when I was marking this, a lot of pupils were just saying, oh, there's a zone of inhibition. Great, you've described that to me, but you then have to tell me what that means. Okay, so that's what the explain means. So the explain is there is a zone of inhibition. Why does that prove that the student is not correct? Okay, so just double check that you have done that in this exam, that you said, oh yeah, there's a zone of inhibition, the bacteria have been killed, there's a clear zone around the antibiotic disc, and which is fine, but that means that if it was resistant, that would not be there because the antibiotic would not be killing the bacteria. Suggest why doctors are concerned about antibiotic resistance. Okay, for 2 marks. All right. So why are we concerned? Because current antibiotics don't kill some bacteria, or they have no effect. Okay, so particularly in the news at the moment, there's loads of strains, particularly sexually transmitted infections. There's loads of strains of that are resistant to all known strains of antibiotic, which is really quite stressful. Why is this a concern? It means that diseases are going to become more common, or can't be cured. Okay, or more people are going to become ill. Some diseases become more difficult to treat. That is all the same kind of mark point idea. Okay, and then finally, you could say new antibiotics take time to develop. So that's why we should be concerned, because it could be that there's a time where we have no antibiotics. You could also mention it takes lots of money to develop as well. Okay. Diseases caused by virus cannot be treated using antibiotics. Suggests why viruses cannot be grown on agar. Now, this is one of those where it's if you don't say a certain word, you cannot get the answer. Okay. Now, and this is because viruses need living cells to survive or reproduce or exist. Okay, so the words that I put in capital letter there, need, you have to say that. Now, you could alternatively say viruses only exist inside living cells. Agar is non-living. Okay, so you need to say, now another thing you could say is viruses reproduce inside living cells and agar is not made of cells. Okay, is another thing that you could say. All right, but again, it's this idea of agar is non-living, so viruses cannot be grown on that because viruses need living cells. Okay, so there's a couple of ways that you can say that. Viruses need living cells to survive or reproduce. Viruses only, again, only, if you just put viruses exist in living cells, right, and okay, why does that mean they can't be grown on agar? See what the issue is. Okay, you need to make sure that it's like, well, they need it or they can only be inside. If you are going to say that viruses need living cells and agar is not made of living cells, that's absolutely fine. Okay, why is it difficult for scientists to develop drugs to destroy viruses? It's because viruses, well, there's three different things you could say. I would go for they frequently mutate. That would be mine. Okay, they do frequently mutate. Another one is that because viruses exist inside of body cells, the drugs would destroy the body cell to get to the virus. Okay, so we don't want to do that. So it's hard to get drugs into. So it's hard to get drugs into living cells, is one. And another is drugs can damage the living tissues or damage the cells because viruses live inside them. Okay. Which disease is caused by a virus that damages white blood cells? Only one answer there, which is AIDS, acquired immune deficiency syndrome. So remember that people who suffer from HIV/AIDS don't die from HIV/AIDS. They die from being immunocompromised. So they die from cancer, they die from fungal infections, they die from bacterial infections, they die from another type of viral infection. Okay, gonorrhea is a sexually transmitted bacterial disease. Measles is a droplet infection virus. And salmonella is obviously food poisoning. Okay, so the only thing that damages white blood cells there is AIDS. Right, so here we've got a student investigating the effect of different factors in photosynthesis. The student used 3 leaves growing on the same plant. Each leaf was treated in a slightly different way, and after 48 hours, the student tested each leaf for starch, and table 3 shows results. So leaf 1, okay, upper and lower surfaces covered in black paper, there was no starch present. Leaf 2, upper and lower surfaces covered and sealed with transparent plastic, no starch present. Leaf 3, not covered at all, and there was starch present. Now the question is explain the results for the 3 leaves. So you have to explain what is happening at 1, what is happening at 2, and what is happening at 3. Okay, if you don't do that, you are not going to get all 5 marks. Okay, so number 1, covered with black paper. Okay, it's covered with black paper, no light, equals no photosynthesis. Okay, now if there's no photosynthesis, there's no glucose made, okay, and then there's no starch stored. Or no starch, no glucose made, so we've got no conversion to starch. Okay, right, number two, right, number two, we've got light this time, but because it has been sealed, okay, there's no CO2, which equals no photosynthesis. Okay, which means no glucose, which means no conversion to starch. Okay, and then finally, three, light and CO2 present, okay, which equals photosynthesis. Okay, now I have done something extra here that you do not have to, but only for either 1 or 2, as long as you have mentioned this once, you will get all 5 marks. If you have not mentioned that, you will get a maximum of 3 marks. So if you have said there's no light, there's no photosynthesis, there's no CO2, there's no photosynthesis, there is light and CO2, so there is photosynthesis, that gets you 3 marks, okay? Because you have not explained this bit, if you have not mentioned that there, okay? So, you have to make sure that you are linking the ability for a plant to photosynthesize with whether starch is present or not, okay? However, now you might have said for leaf 3, if you have said it can photosynthesize, so it can make glucose and can be converted to starch for storage, you have all 5 marks, okay? So you either say there's no glucose made and there's no conversion to starch for 1 or 2, or if you've done both, well done you, okay? Or you get your 4th and your 5th mark from saying the 3, light and CO2 is present, so it can photosynthesize, so it can make glucose, so it can convert it to starch. All right, so those are your other options there. In another investigation, the student used a different type of plant. The plant was left uncovered in light for 48 hours. After 48 hours, the student tested a leaf from the plant for starch. Figure 6 shows the leaf before it was tested for starch. So this is something called a variegated leaf. Okay, now variegated leaves are the ones that look really pretty because they're kind of like white around the edges and they've got a little bit of green on it, or they're kind of green and white, and they've been bred to look that way because they look quite pretty. Okay, so the question is, complete table 4 to show the results you would expect for the starch test on the leaf in figure 6. So it was left uncovered, okay, completely uncovered. So green, I would expect to see starch present because it's green, it contains chlorophyll. Okay, if you've got chlorophyll and we're uncovered, it means we've got lots of light and CO2, we're going to get starch present. Okay, now alternatively, you may interpret that because it's asked for the starch test, you may have said blue-black. Okay, that is also fine. If you've written blue-black, or you've written starch present, or you've written both, congratulations, that is fine. Okay, I will also allow you to say positive for starch as well. Okay, now the white, there is no chlorophyll. So if there's no chlorophyll, all right, it cannot photosynthesize. So there would be no starch present, or you would say it would remain orange or yellow, or whatever it is you want to say. Orange, yellow, even brown will be accepted as a colour. Explain the results you gave in question 7.2. Okay, so we're explaining those results that we've just written. Okay, and I've just told you essentially. Okay, so the green contains chlorophyll, white does not. Now here's the thing, if you have said chloroplasts, no dice. You have to say chlorophyll, okay? Really particular. So it's not the chloroplast the chloroplast houses the chlorophyll, but it's the chlorophyll that is required for photosynthesis. Okay, so then we say, so light is absorbed by green part, or you can say is absorbed by the chlorophyll, so photosynthesis can occur. So photosynthesis occurs and starch forms. Now, to be fair, that is a lot of work for 2 marks. I think that's a bit stingy, but there we are. So you need to say green part contains chlorophyll and the white part does not to get your first mark. Your second mark, light is absorbed by the green part, so photosynthesis can occur in starch forms. Okay, because again, you need to explain the results. Why is it positive for starch? Why is it negative for starch? In some leaves, the green parts become yellow because of an ion deficiency. Which ion is deficient in a plant with yellow leaves? Okay, so it is magnesium. That is, now, to be fair, it will also allow nitrate, not nitrogen, okay? Or if you are going to be clever, right, it's Mg, like that. If it's not like that, if you put capital M, capital G, no. And if you are going to do the ion and actually do the actual symbol for it, it's 2 plus. Anything other than 2 plus, it is not it. Okay, so just be a reminder of that. What name is given to the term that describes the yellow colour of the leaf? There's only one thing that we can say here, and that is chlorosis. The rate of photosynthesis is affected by different factors. How could the oxygen produced during photosynthesis be used to measure the rate? Okay, so again, it's asking you to measure the rate. It's highlighted, it's bolded the word rate. Now, if you want to know the rate of something, you need how much is produced and you need a time frame. Okay, so you need both of these things mentioned in order to get the 1 mark. So it's the volume in a given time. Or you could say the volume of oxygen released in 5 minutes or the volume of oxygen released in 1 minute. That's fine. So if you've not actually said a given time and actually given me a time, as in 30 seconds, 1 minute, whatever, that's fine. Congratulations, have a mark. But you have to say the volume released in that time or the number of bubbles in a given time. Okay? So the volume or the number of bubbles. Okay? Fab. Light, carbon dioxide, and temperature are limiting factors of photosynthesis. Figure 7 shows how the rate of photosynthesis is affected by light, CO2, and temperature. At point A on figure 7, light is a limiting factor. What is meant by limiting factor? So it's a factor that prevents the rate increasing. Factor that prevents the rate increasing. You could also say that if increased would increase the rate. Okay, so if increased would increase the rate. Okay, you can also say it's a factor that prevents the maximum rate being reached is another one as well. Okay, so there's a few things that you could say there. You could also talk about it in terms of a named factor. So, for example, you could say if water wasn't present, then that could prevent the rate from increasing. That's fine. So instead of saying factor, you could say water, CO2, temperature, whatever. Okay, maybe not. No, yeah, maybe temperature as well. Explain the effect of increasing temperature and increasing CO2 concentration on the rate of photosynthesis shown. Okay, right. So this is a tricky one. Okay, so what we're looking at here is this here. We've got the same CO2, but then we've got an increase in temperature. Same CO2, increase in temperature. So what we need to say is that increasing the temperature while keeping the CO2 constant increases the rate. So increasing CO2, sorry, not increasing CO2, increasing temperature whilst keeping CO2 constant increases the rate. Now that's just 1 mark. Okay, so there's quite a lot that you need to write in order to get the 4 marks here. I think, again, it's a bit of a mean question. Okay, increasing temperature, and now we need to, because we're explaining it, we now need to say, why does increasing the temperature have an effect? So this is essentially, collision theory. Okay, so increasing temperature increases the movement or the kinetic energy of the particles. So it increases the frequency of collisions. Okay. And then increasing CO2 increases the concentration of the substrate or reactant. Okay. Now, what then we need to say is the final, so that's 3 marks. And then the final thing we need to say is all rates plateau or flatten, etc., due to another limiting factor. Now, you may name this yourself. So it could be kind of water, or it could be chlorophyll. So that's where your 4 marks are. Increasing temperature while increasing CO2 concentration increases the rate. Sorry, while keeping CO2 constant increases the rate. Increasing the temperature increases the movement and frequency of collisions. Increasing CO2 increases the concentration of the reactant. However, all of the rates plateau at a certain point due to another limiting factor. Okay, so it's a bit of a tricky one, that one. But that's the only thing that you can get. Now, certain things you can say. You could say increasing the CO2 concentration while keeping temperature constant increases the rate of photosynthesis. Absolutely, you could do. So if you look, obviously 15 and 0.04 CO2 and 15 and 0.4 CO2 and 15, there is a big jump, right? So that's another way that you could say 0.1. So instead of saying increasing temperature while keeping CO2 constant increases the rate, you could also say increasing the CO2 whilst increasing the temperature. Sorry. Increasing the CO2 whilst keeping the temperature constant increases the rate. With the increasing temperature increases the movement frequency of collisions, you can say increases the movement, increases the kinetic energy, or increasing the temperature increases the rate of enzyme activity. You can have that as well. And then increasing CO2 increases the concentration of reactant or substrate. That's the only thing you can say there. And then you could say all rates plateau due to another limiting factor, or you could name it. So, for example, water or chlorophyll. Okay, there we go. This one, photosynthesis investigations often use a light source. The spreading out of light from a source obeys the inverse square law. The inverse square law links light intensity to distance. Which of the following shows the inverse square law? And it is this one here. Okay, so that's just a pure recall question. Cancer is caused by changes in cells that result in uncontrolled cell division. Before a cell begins to divide, before it even starts to enter mitosis, its DNA replicates to form 2 copies of each chromosome. One other change. Okay, now you can say increase in subcellular structures. That's fine. Increase in mitochondria, increase in ribosomes, increase in cell in respiration, increase in protein synthesis. That's it. Okay, so we're looking at increase in the number of subcellular structures. That's kind of like a catch-all. Or you could specifically say increase in number of mitochondria or increase in number of ribosomes. Or increasing amount of respiration. Or increase in respiration rate. Okay, so those are your options there. Anything with talking about the nucleus, anything with increasing cell parts or cell components, not scientifically accurate enough. And any changes that occur as the cell divides, so anything to do with lining up or anything, no. Okay, figure 8 shows a cell during one of the stages of cell division. Okay, it's pretty obvious which one this is. It's when they are being pulled to opposite ends of the cell. Name structure Z in figure 8. So this is just the membrane. Okay, you don't have to put cell. If you've put cell membrane, absolutely fine. If you've just written membrane, absolutely fine. If you've written membrane, membrain, that is fine. Phonetic spelling is okay. Okay, so this is a bit of a long-winded one, all right, but it's a good one if you get your head around it. Structure Y in figure 8 is a cylinder. For structure Y, the real volume is 24,500,000 nanometers cubed. The real radius is 125 nanometers. The length of a cylinder is calculated using the equation length equals volume divided by pi times radius squared. And it's told us to use 3.14 as pi. Now, it's then given us the length is 4 millimeters in real life, and then we've got to calculate the magnification. Sorry, it's 4 millimeters on the image, and it's asked us to work out the magnification. Okay, so this length here that we're going to work out is the length in real life. Okay, so first thing that we've not got included on here that we're going to have to use is our AIM equation. Okay, so first thing I'm going to do before I start thinking about the magnification is I'm going to work out what the length is in real life. What is the actual length? Okay, so length equals the volume, okay, which is 24,500,000 divided by 3.14 times 125 squared, and that comes out at 499.363. Okay, nanometers. All right. I've got my magnification is image over actual. Okay, I've got my image is 499 nanometers, but then I've got my actual is 499 nanometers, but my length is in millimeters. So that needs converting into nanometers. Now, there are 1,000 micrometers in a millimeter, and then there are 1,000 nanometers in a micrometer. So it's going to be 4 times 1,000 gives us 4,000, and then times 1,000 again. So that gives us 4 million. Okay, so it's going to be image is 4 million divided by and then we're going to divide that by 499.363, and that comes out at 8,010. So that there is my answer. Okay, now you may have rounded it, so it'd be 8. If you've rounded it, and got rid of that bit and just said 499, you'll get 8,010. If you've not, it'll be 8,010.205, something like that. Okay. So the issue is have you done that? Okay, because if you've not done that, you ain't going to get the full marks there. Okay, so you get 1 mark for your substitution. Okay, 1 mark for the length, 1 mark for the AIM equation, 1 mark for the conversion, and then 1 mark for the answer. Okay. If you've just written the answer and you've not done any other working, you will still get 5 marks because that's how biology works. Okay. Figure 9 shows some of the stages of cell division. So we've got stage 1, 2, and 3. Stage 1, fibers attached to replicated chromosomes. Stage 2, and then stage 3, fibers attached to the chromosomes get shorter. Some cancer drugs prevent cell division. Drug X prevents the fibers from attaching to the replicated chromosome in stage 1. Explain why a cell cannot complete division when affected by drug X. So the chromosomes can't be pulled to the end of cells, or opposite ends of cells, cannot be pulled to opposite ends of cell. Which is a lot again for one, that's again 1 mark. And the other mark is, so nucleus cannot divide. Now, there is a couple of other ways that you can say this. So you could say 2 genetically identical cells cannot be formed. That's it. Anything, if you've mentioned cytokinesis can't happen, no. If you've mentioned the cell cannot divide, you can't get that out. If you said the cell cannot divide, because it's already told you that the cell can't divide, so you need to tell me why. Well, it's because the nucleus isn't able to divide. Okay. 8.5, give the reason why a drug that stops cell division helps to treat cancer. So it's essentially that the tumour can't grow or spread. So tumours cannot grow or spread, or you could say metastasized, or stops metastasis, metastasis, okay, or stops secondary tumours from forming. Right. There we are. And then new cancer drugs are tested in clinical trials. Preclinical trials happen before clinical trials. What is involved in preclinical testing of drugs? And it's testing it on live tissues in a laboratory. Okay, so that is the end of your separate sciences walkthrough. If you've got any questions on anything that I've said, please do come and find me.